Hello, sir. This is Yash Agarwal here, along with my team members. So, we are here to present on the topic dialysis industrial application. Now, here are the contents in which we will first introduce the topic and then we will carry forward the presentation with its applications and examples, and then finally, we will give a look to some of its advantages and disadvantages. So, now let's start with the introduction. Now, what is dialysis? Dialysis is actually a membrane process by which one can differentiate various solutes having widely different molecular weights which may be separated through semi-permeable membranes by the means of diffusion. Dialysis, it mainly runs on the principle of diffusion, osmosis and ultrafiltration. Industrially, it is also used to correct electrolyte imbalances and also used to remove waste product and excess fluid in the body when the kidney stops functioning properly. Moving forward, let's give a look on the mechanism and the process of dialysis. Now, as we can see from the figure, a biological sample is placed inside a tube of semi-permeable membrane and the whole sample is placed inside a much bigger container containing buffer. Now, as the time passes, only small molecules whose size is smaller than the pore size of membrane diffuse through the colloidal membrane. Once, when the equilibrium is achieved across the entire solution, the concentration of small molecules becomes same inside and outside the membrane. Further, if we replace the equilibrated dialysate by a fresh dialysate, then further diffusion will occur which will reduce the concentration of small molecules in the sample and the larger molecules will remains in the bag. Now let's give a look to the video. So here we have seen in the video on how the process of dialysis works.
Hello sir, I am Samyak Jain. So now we are going to look into some of the applications of dialysis. So the first one is the removal of salts and low molecular weight compounds. Dialysis facilitates the removal of small, low molecular weight compounds from larger molecules in solution by selective and passive diffusion through a semi-permeable membrane. Initially, a sample and a buffer solution are placed on opposite sides of the membrane. So sample molecules that are larger than membrane pores are retained on the sample side of the membrane. But on the other hand, small molecules and buffer salts pass freely through the membrane, which will reduce the concentration of those molecules in the sample. Changing the dialysate buffer removes the small molecules that are no longer in the sample and allows more contaminants to diffuse into dialysate. So in this way, the concentration of small contaminants within the sample can be decreased to acceptable or negligible levels. So now if we talk about removal of salts from larger molecules or macromolecules, which is known as desalting. So desalting occurs when buffer salts and other small molecules are removed from a sample in exchange for water. So for better understanding, let us give a look on this video. this is how the removal of salts and low molecular weight compounds from larger molecules through dialysis takes place. Now the next application is the buffer exchange. Buffer exchange occurs when the buffer salts in a sample with which membrane is equilibrated are exchanged for those in another buffer. The next application is the concentration of macromolecules, in which macromolecules like proteins are separated from smaller molecules. And the last one is the purification of biotechnological products. The procedure for last two applications is same as what we have seen in the video.
Hello sir, myself Prajwal. Now let us look into some of advantages of dialysis process. The main advantage of the diffusion dialysis is the low energy consumption of unit as there is no phase change. Also, considering about nature, diffusion dialysis does not pollute the environment. On the other hand, talking about that of benefits of the electrodialysis is its high recovery, especially in the water recovery. Another advantage is fact that high pressure is not applied, which implies that the effect fouling is not significant and consequently no chemicals are required to fight against them, which leads to a higher recovery and to a long membrane life. At last but not the least, there is low installation cost, easy and cheap operation, and the process is stable and reliable. And now, moving forward, let us look into some of its limitations. A diffusion dialysis has a low processing capability and low processing efficiency, which makes it less preferable over electrodialysis and reverse osmosis, which can achieve better efficiencies. On the other hand, the main drawback of electrodialysis is the limit on current density, due to which the process must be operated at a lower current density than the maximum allowed. Also, in electrodialysis, a post treatment is necessary as it is considered that the process does not remove microorganisms and organic contaminants. Also, the slow process of dialysis takes several hours for completion, and thus it has been replaced by gel filtration for most of the applications. So, this is all from our side, sir. Thank you so much.